Everybody. This lecture now begins our exploration of large biomolecules. The first one that we're going to look at is carbohydrates, or our carbohydrates. When we look at energy and metabolism with our last lecture in a week or so, what we're going to see, and what you might even remember from some of your basic high school biology, is that the photosynthesis and cellular respiration sort of cyclic pathway centers in on glucose and the production of glucose by photosynthesis, and then the breakdown of glucose by cellular respiration. And things like starches and other complex sugars, all part of that big process for how we get energy in this sort of cycle of life, so to speak. And so to really start to understand some of that a little bit more, we need to look at that basic building block of glucose, which is a carbohydrate. Almost half of all of the carbon on Earth exists in a carbohydrate form. And so, as you know, just from your some basic science and basic biology, plants produce carbohydrates and oxygen from carbon dioxide, and that's really what photosynthesis is all about. We human-centric people, I guess, we view photosynthesis as, oh, this is how we get oxygen. From the plant's point of view, though, the entire purpose of photosynthesis is this production of carbohydrates, this taking in of carbon dioxide and water and using those as the components to build sugars. And so really, oxygen is just a byproduct. It's a lucky byproduct for us, but that's all oxygen is from the plant's point of view, just a waste product that it gets rid of. When starch, a very complex carbohydrate, is consumed, it ends up being metabolized and broken down into glucose. The whole process begins with an enzyme present in our saliva and then continues through our small intestines. But that's one of the purposes, really, of our entire metabolic process, is being able to convert some of the foods that we eat into glucose, which is a carbohydrate. And then that is used in some of the pathways that we'll talk about later in order to get cellular energy. So the carbohydrates then provide fuel for cells. They provide building blocks for things like DNA and RNA, and they're very important for different cellular recognition processes. The basic structure of all carbohydrates fit the molecular formula of CH2 and then N. And so any sort of number or any sort of maybe repeating chain of that basic formula. And so as a result of that, it was theorized early on that these molecules were actually what we call hydrates of carbon. Carbon hooked to water molecules. We found since that that's not true, that these are actually polyols. They're compounds that have multiple alcohols on them. However, the name remained. And so we stuck with the name of carbohydrates. When we start to look at carbohydrates, one of the other names for them are saccharides. And that name maybe sounds familiar as sugars, saccharide, sugar. A monosaccharide means that it's made up of, just as the name implies, one saccharide unit. Some of the identifying features of different types of monosaccharides are, one, the number of carbon atoms in the chain, and we'll be talking about this later on. There's also a carbonyl group of some sort on monosaccharides. And the type of carbonyl that's present is another identifier of the type of monosaccharide we're talking about. And it will be either an aldehyde or a ketone. Another important identifier is what we call the stereochemical configuration. And we're going to look at that a little bit later in this lecture, but not in a lot of detail. Most common monosaccharides, rather, are straight chains of anywhere from 3 to 6 carbon atoms that have an aldehyde at C1 or a ketone at C2, and then a hydroxyl, an alcohol group, hanging off all other carbons. And so if we just look at a random example, And so again, straight chains of 3 to 6 carbon atoms. And so we could have 1, 2, 3, 4, 5, 6, maybe 3 carbons there. And we could have possibly an aldehyde at C1 or a ketone at C2. And so let's say we have maybe an aldehyde here at C1. So here's C1. And so we would have a double bond O with our H coming off it there. So there's an aldehyde. And then all other carbons would have an OH group coming off. And so this would be some sort of generic saccharide, some sort of generic carbohydrate molecule. And some of the different features of this, or the fact that one, like I mentioned, that we have an aldehyde here at C1 as opposed to a ketone at C2. And also the way that these OHs are actually arranged. Some of them might be arranged coming out at you. Some of them might be arranged coming back. Thinking about our Vesper shapes that we talked about earlier on with that three-dimensional shape. That's what that stereochemical configuration is all about. Which direction are those OHs facing? Because that will make a difference as well. But as just a generic hydrocarbon, this is a good depiction of one. So at least a 3-chain carbon atom, anywhere from 3 to 6, or sorry, 3 to 6 chain carbon molecule with an aldehyde or a ketone, aldehyde at C1, ketone at C2, and hydroxyls on all other carbons. This would be a very generic picture of a hydrocarbon here, sorry, of a or carbohydrate here. Most carbohydrates, the name ends in ose, and some that you're familiar with, things like glucose, fructose, sucrose, and so on. Monosaccharides, what we call simple sugars, cannot be hydrolyzed into smaller units. Hydrolyzed is just a way of breaking them up. And so monosaccharides cannot be broken down further and still be a saccharide. I mean, yes, you might be able to break up the whole chain, break it into its constituent atoms somehow, but it wouldn't be a saccharide. It wouldn't still be a sugar of some sort. Disaccharides, however, can be hydrolyzed, can be split up into 2 different monosaccharides. And we're going to see some examples of these later on. Oligosaccharides and then polysaccharides just get larger and larger and larger, but they could all be broken down into monosaccharides. You can picture a monosaccharide essentially as being sort of a carbohydrate building block. You could have just one, or you could have various chains of them, either the same type or different types. And we'll take a look at some of that. And so chirality is something that we haven't talked about yet. Let's look at just a quick overview of what chirality is. So if we have some generic molecule here, maybe an OH right here, and then if we think about the 3 dimensions around that OH, and we've looked at some of these depictions earlier. Let's say we depict this as this OH is coming out at us, which means remember that the hydrogen written as a dash would be going away. So again, remembering the 3 dimensions of that, this would mean that the carbon chain is sort of all in the same plane. The OH, let me redraw that a little bit clearer. The OH then would be coming out of the screen, coming out towards you, and the hydrogen would be going back into the screen. Now, it turns out that even though everything's pretty much connected the same, that if we reverse that, if instead we say that the OH is going back and the hydrogen is coming at you, these molecules would exhibit a different, what we call chirality. And we're not going to go much deeper than that, except for knowing that this different configuration like this could give the molecule different properties. We can also look at this as what we call, they are non-superimposable mirror images of each other. An example of that is your hands. Your hands are mirror images of each other. And so my 2 hands here are mirror images of each other, but they're non-superimposable. If I put my hands in the same configuration, both with palms down, and I place them on top of each other, my thumbs don't line up. for instance, and the fingers don't all line up with each other. And that's the same thing, that's the same principle as chirality, a non-superimposable mirror image. And so we have a number of these, what we call chiral centers in carbohydrates that have a different, what we call stereochemical configuration, which means that the atoms and some of the different OH groups, for instance, are either coming out or going in, and they're taking up different areas in space. And that's one of the key differences between some of the different sugars. Might be literally a difference between an OH pointing up or pointing down, but that would be a key difference in identifying a different type of saccharide. And so quite often, in order to depict this, we draw a saccharide, we draw a sugar as what we call a Fischer projection, and that's what you see here. And this helps us to differentiate between two different, what we call enantiomers of the same type of sugar. And so here's a Fischer projection of a glyceraldehyde. And we can see here that either the OH is pointing to the left, that would be the type of glyceraldehyde we call L-glyceraldehyde. And here this OH is pointing to the right. And so that's D-glyceraldehyde. When we're drawing these Fischer projections, the carbonyl, so remember when we're looking at I can't think of words today. When we're thinking of the different properties of saccharides, different properties of these carbohydrates, there's always either that ketone at C2 or the aldehyde at C1. Those are carbonyl groups. And so when we're drawing a Fischer projection, that carbonyl group is always up here at the top. That's what this CHO is. That indicates an aldehyde. And we then, so the carbonyl is at the top, and then each hydroxyl group, each OH, is placed on a line like this. And so these are these lines that you see right here. And this is a very simple, glyceraldehyde is a very simple saccharide. But these OHs here, these are the same as, come back to here, is this sort of simple one that I drew here. And so each of those vertexes would essentially be like these carbons here, would be those with the OH. Because remember, there's also an H coming off each of those. It's just not drawn in there. And so that's what each of these little intersections are here. Carbon atoms that are not chiral are written in this condensed notation, and that's what this is down here. In order to be chiral, there have to be 4 different things coming off the carbon, which we can see that here. This carbon here has 1, 2, 3, 4 distinct groups, making this a chiral carbon. So again, we're not going to worry too much about that. But what you do have to take from this Fischer projection is that when they're drawn like this, I'm just looking ahead. Yeah, actually, I'm going to talk about that a little more later. So don't worry about that too much now. Here's a nice drawing again of the difference, and you can really see it here. So here's a ball and stick model of glyceraldehyde. And we can see D-glyceraldehyde here. So here's that OH coming out at you, coming out of the screen. And then here it is flipped over. So we've taken glyceraldehyde here and flipped it. And so now that we flipped it, the OH is still coming out at us, but the whole thing is flipped over. So that's L-glyceraldehyde. If we kept it in the same sort of configuration with our carbonyl here at the left, then in that case, the OH would be going away from us. And so those are 2 different enantiomers of each other. D monosaccharides, again, will always have that hydroxyl group written on the right. L monosaccharides will have that hydroxyl group on the left. And there's a certain hydroxyl group that we'll look at, and we'll talk about that. Most monosaccharides do have multiple of these chiral centers. And so the number of possible isomers that could exist would be 2 to the n, where n is the number of chiral centers. And so in this case here, we only have 1, just 1 center right here. And so 2 to the first is 2. So there are 2 possible configurations of this. We're not going to do too much about that other terminology. So again, these different enantiomers, these different forms of the saccharide are identified either as D or L. We're not going to worry about the diastereomer aspect. In nature, only D-type sugars are actually produced, and that's important to know. In a D sugar, the O, and this is the key aspect. You're looking at the OH group that's at the intersection farthest from the carbonyl. In an OH group, it's the intersection farthest. That OH group at the intersection farthest from the carbonyl is on the left. So here's an example here of glucose. So again, here's our Fischer projection. Here's our carbonyl at the top. This is our aldehyde, our CHO group. And we can see that we've got a number of different carbons. It's a 6-carbon chain here. But we can see that while we're looking at our carbonyl here at the top, we've got this OH that's the furthest from the carbon. Now we're looking, some of you might be saying, well, Professor Staples, this one's actually furthest from the carbon. But see how this one's written in the condensed form? That means that this one's not our chiral center. We're looking for at these intersections, for lack of a better way to put it. So we're looking at all these intersections here, and this is the intersection in our Fischer projection furthest from our carbonyl. And so we see the OH on the right. That means this is D-glucose. The OH on the left, it means that this one is L-glucose. As mentioned earlier, monosaccharides, remember, can have either an aldehyde at C1 or a ketone at C2. And so we have our Fischer projections, which we've looked at. It turns out that in nature, monosaccharides that have 5 or more carbons are actually going to take that chain and fold around in order to form a ring. And we can see that happening right here. And so what's going to happen is that a carbon-oxygen single bond will form between this last chiral carbon here, so here at number 5, and this oxygen of the carbonyl. And so we can see that sort of as it comes around here. So here's that carbonyl group. So right here is this. And then what's going to happen is that the OH, this O right here, is going to then form a bond. And that will form our ring. And we're going to talk about alpha and beta, what these mean in just a minute. But this gets us to our ring structure of our saccharide, which is sort of the common form that we think of saccharides to be in. And here's a much better blown up picture of it. So again, we can see our Fischer projection here in our chain. And what's going to happen is we're going to form a new bond. Here's our OH. That's that's the alcohol on our chiral center furthest away from our carbonyl. And so this OH is the same as this OH right here. And this carbon is going to come around and then form a bond right there to form the ring. And some other things happen here too. This H obviously goes away. This O here ends up becoming an OH group right here. And we're not going to worry about the chemistry of really what's going on there and how that occurs. Just know that that is what ends up happening. And then here at this 1 position, this is now what we call the anomeric carbon. And that's a piece of terminology that we're going to be using here in just a little bit. But at this 1 position from our ring, we call that the anomeric carbon. A couple other sort of definitions to know about. A 6-membered ring that is formed is called a pyranose ring. A 5-membered ring is called the furanose ring. When the rings are formed, we now form a new chiral center, gets formed around that carbonyl carbon. And so, like I said, that is what we know, or what we call the anomeric center, or the anomeric carbon. Then what's going to happen is that oxygen that was part of the carbonyl becomes an OH group. If we think of the ring as being sort of the plane of my hand, that OH group might be above the ring or it might be hanging below the ring. And believe it or not, that matters. And so that ends up getting different terminology as well. If the OH group is below the ring, we call that the alpha version of the saccharide. If that OH group is above the ring, that's called the beta version. all three forms, when we go from the chain into our ring, all three forms, the alpha, beta, and a bit of the straight chain, are actually present. One of them usually in either the alpha or the beta in a higher proportion, but a little of both. And again, so we can see a nice depiction of that here. Here's our Fischer projection again of glucose. So remember that this OH right here ends up forming a bond to this carbon. So here's our new bond that's formed. So here's our anomeric carbon. And we can see here that at the anomeric carbon, our OH group is below the ring. So that's the alpha version. And so this would be alpha delta glucose, or alpha D glucose. Sorry, it's not delta. And we knew that it was D because our OH group right here, remember, is on the right, making this the D glucose form. And then we have beta D glucose because our OH group is up above. We'll quite often also draw monosaccharide rings as what we call Haworth projections. And you can see that here. So here's a Haworth projection of these two different saccharides. And you can see that this is a little bit more shaded. This is to indicate that this is coming out at us, whereas the rest of this is sort of going back. And so we're picturing it as a plane like this. And what the way that we sort of interpret or go from a Fischer projection to this Haworth projection is that groups that appeared on the right in a Fischer projection end up below the ring in a Haworth projection. Groups that were on the left in a Fischer projection end up appearing above the ring in a Haworth. When we're looking at the differences between a furanose or a pyranose, the oxygen is always at the top for our furanose ring. So here's our oxygen right at the top of the furanose ring. And the oxygen is always in the top right for a pyranose. And so those are a couple of conventions that we always use when we're going from the Fischer to these Haworth projections. Pyranose ring, the oxygen will always be in this location of the hexagon. In a furanose ring, it's always right at the top. And then this is the key, or one of the keys, when you're looking at these Haworth projections of carbohydrates, your anomeric carbon is always going to be the one directly to the right of the oxygen. And so when you're looking to try to identify, is this particular saccharide an alpha or a beta, that's what you want to look at. And so if we draw a very generalized pyranose ring, so here's a very general pyranose ring. It wouldn't matter. So again, remember this, and this one of the defining aspects of saccharides is that there's going to be OHs hanging off each and every carbon. If we're trying to determine alpha or beta, we need to look at this one right here. And so if our OH is below the ring, that's our alpha version. If the OH happen to be above the ring, everything else is the same. Our OH now is above the ring, then that's our beta version. But that's where we're looking, is that anomeric carbon, which is always going to be the one to the right of the oxygen. And so just another depiction of what's going on there. There's our Fischer projection over to our Haworth projection, beta with our OH going up, and alpha with our OH coming down. And we can see again that we see here, like for instance, at the 2 position, our OH is to the right, and so that points down. OH to the left points up. Disaccharides are combinations of 2 different monosaccharides, and that should make sense. And so if we take 2 monosaccharides and we join them in what we call a glycosidic bond, we'll end up with a disaccharide. A glycosidic bond occurs between the anomeric carbon on one of the monosaccharides and one of the hydroxyl groups on the other. And so here we have galactose. And I should back up. The different types of sugars, so if we have different types of monosaccharides, the difference is where these OHs are pointing. These three different OHs here. And I don't remember, there's so many different types, I don't remember the names of all of them. But if we had just, if this hydroxyl group at carbon 4 were pointed down, this would be a totally different sugar. And so that's the defining aspect of different carbohydrates is, are these sugars, or one of them, are these hydroxyl groups pointing up or down? So anyway, here we have D-galactose. So this is a galactose sugar right here. And we can see our anomeric carbon. And so then we have a glucose molecule. And we can see that the OH, the hydroxyl group here at the 4th position, is now forming a bond. So that O forms the bond to our anomeric carbon on the galactose. That now links these two together into a disaccharide. And this, in fact, is a unit of lactose. People who are lactose intolerant, this is the bane of your existence. This molecule here is why you can't enjoy ice cream. And I feel sorry for you if any of you fall into that category. Ben and Jerry's Chubby Hubby, absolutely delicious. Sorry to rub that in. But let's look closer at that glycosidic bond. We have different types. The most common types are beta 1,4 and alpha 1,4. And let's look at the differences there. The beta or the alpha, that just depends on the saccharide that is being joined at the anomeric center. Did that start as an alpha or a beta? Simple as that. 1,4 tells you which carbons are actually bonding together. And so if we look at some examples here, and so the first number will pretty much, I'm trying to think if there are any differences. I think will always be 1 because that's our anomeric carbon. So here's the one that we were just looking at. You might need to zoom in on your screen a little bit, or maybe look at this in the lecture slides so you can really zoom in and see it. But here we have the saccharide lactose, so again made up of a galactose and a glucose molecule. Here's our glucose molecule here, and we know that this was beta glucose because our OH is pointing up. So here's our O pointing up, so it's beta. It's connected at the 1 on glucose, and it's connected at the 1, 2, 3, 4th carbon of glucose, hence beta 1,4. Here's a monomer of sucrose. So this here is a sucrose molecule, common table sugar, which is made up of glucose and fructose. So here's our glucose here. This is an alpha 1 beta 2 linkage. Now this gets a little bit different just because we're looking at a 5-membered ring here, which gets numbered a little bit differently. And so your anomeric carbon on 5-membered rings is actually the number 2 carbon. But if we look at this, we can see that it's alpha on the glucose because our OH is pointing down, and then beta on the fructose because the OH was pointing up. So we get what we call an alpha 1 beta 2 linkage. Cellobiose, made up of 2 different glucose molecules, beta 1,4, beta because we're pointing up, 1,4 because we're from the 1 on this glucose to the 4 on this glucose. Same thing here, maltose is also made up of 2 glucose molecules, also connected at 1,4, but now this is an alpha linkage because it was coming down. The OH was coming down off the ring in this glucose molecule. So very different sugar. And the key difference is basically that it's the alpha versus the beta. And so in the beta, we end up with a linkage that's sort of like this. So 1 molecule here and 1 molecule here. So they're kind of stacked a little. Whereas in the maltose, they can be more in the same plane because that alpha linkage comes down below. And so different linkages can lead to those different sorts of configurations. So here's just another example of that. So here's our alpha 1 beta 2 between glucose and fructose, and we've already seen that. We've looked at it already, so I won't spend much more time there. And so that's how a disaccharide can form. Polysaccharides are what we call biological polymers. And so now we're going to get long strings of usually glucose monomers connected in either alpha 1,4 or beta 1,4 linkages, and a few different types of those: amylose, amylopectin, cellulose, glycogen. This is how plants store energy. Plants store energy in the form of starch, which is 20% amylose and 80% amylopectin, just long strings of glucose molecules. And we'll look at a couple diagrams of that. Mammals store their glucose as glycogen. And so if we take in too much glucose, more than our cells need, we're going to then store that as glycogen, which is going to be just glucose molecules. So picture just like one glucose ring, now linked together, anywhere from 250 to 4,000 molecules long. That's glycogen. Or that's one of the various polymers. Amylose, unbranched polysaccharide with a helical shape, and there's some good diagrams of this, I think, in your book. Let me take a peek real quick. What chapter are we in here? Chapter 14, yeah. And we'll see some pictures here in a second too. But if you look on page 550 in your book, you see some good diagrams there. Yeah, there we go. That's the same diagram I'm looking at here. So there we have amylose. So you can see the little monomers right here. So there's a sugar, there's a sugar, there's a sugar, and they're all sort of connected in that helical shape. Amylopectin, a branched polysaccharide with an alpha 1,6 linkage, ends up with a spherical shape. So here's what amylopectin would look like. And then glycogen and cellulose. So again, really one of the takeaways you need to take from this is that all these different long polysaccharides are just sugar molecules in long extended chains connected in different ways. And they'll have different sorts of, they have different sorts of purposes. Starch is the primary storage form of glucose in plants and made up of mostly amylose and amylopectin. Glycogen is how animals store their glucose. And then we have cellulose, which is basically the structural role, the structure of plants. So plant stems made out of cellulose. And so again, remember, way back to photosynthesis, that's the whole point of photosynthesis from a plant's point of view, is to make these little glucose molecules here so that it can string them together and make larger, more complex structures. Most cell markers, or some of the various things bonded to cells, many of those are made up of oligosaccharides, longer sugar molecules. A great example here are the 4 different blood types, AB, and O, because they differ in their markers that they have on their cells. People with type O blood have a trisaccharide cell marker. People with type A have a tetraside marker, which includes the 3 from type O. Type B, people with type B blood have a different tetrasaccharide, but that includes also the 3 from O. And AB contains both. Yeah, and so we can see that here. And so people with type O blood, so here's a red blood cell, and it'll have these oligosaccharides hanging off it. And we can see here that they all have this N-acetyl D-glucosamine. Don't worry too much about that, but so this is a saccharide. And so people with type O then also have D-galactose and N-fucose, 2 different saccharides hanging off there. And so all blood types have the same basic chain. People with type O only have that chain. People with type A now also have this other saccharide, N-acetyl D-galactosamine, hanging off it. People with type B have this other type, this D-galactose. It's another D-galactose hanging off this D-galactose. And then people with AB have both of those. And so this leads us to what a lot of you probably know as the sort of the donor and recipient issues. Is the problem is if you take blood, if you're a recipient and you're getting blood that does not, or sorry, you're getting blood that has one of these markers, but your blood doesn't, your body's going to reject that. And so if you are receiving type A blood that has this N-acetyl D-glucosamine, if you're receiving this, but your body doesn't have that, your blood doesn't have that, it's going to reject this blood that you're receiving. This is why people with type O blood can only receive type O blood, because they can't handle, their bodies can't handle these other polysaccharide markers that are on there. It's why people with AB can accept anything, because if they're getting some A or getting some B, they have some of that. It's also why type O can donate to anyone, because it's not giving out any of that extra marker. And so here's a sort of a real world example of where some of these polysaccharides and oligosaccharides come into play. Is they are the main determining, they are basically what determine different blood types. And so, okay, I know that sort of and I should have mentioned this in the beginning of the lecture, but our format is changing a little bit, in that these last few lectures aren't so much process type things like we've done before, learning how to balance equations and show you examples, to do calculations, to show you how to name organic molecules. These next few lectures are basically information dumps, where I talk about a lot of the things that are in the book, but it's a lot of just, here's all the information. And so that is a lot to take in. But there aren't really good ways to break some of these up other than in the main categories like I'm doing. It does make for longer videos, but hopefully you've been able to bear with it. If you took a break here and there through it, I definitely do not blame you at all. But that is it for our carbohydrate video. And so until next time, you stay classy, USM.